morning again. Welcome back, everyone. I hope you all had a very good lunch. So I'm going to start the afternoon with a talk about consensus modules. What it means is really modules present across multiple data sets. And the reference for our work is down here. Let's see if I can get the mouse pointer. But I'll post the reference one more time. So why would you want to consider consensus modules? Again, given a few independent data sets, the idea is to find modules that are present in all, or at least the specified majority of the data sets. And the rationale is really to find co-expression patterns that are common to either the multiple studied conditions or at least multiple independent data sets that study the same condition. And so you want to find common, robustly defined modules for data that study either the same or the similar condition. How do we find consensus modules? Well, you have heard in the morning that modules are groups of genes that are tightly or densely interconnected. And so consensus modules then are a group of genes that are densely connected across all of the studied data sets. So the solution to finding consensus modules is to find the consensus gene-gene similarity or dissimilarity and then use it with clustering to find modules. So here's a cartoon. You start, let's say, with two networks, network 1 and network 2. And I'm representing the networks by the heat map. So what you should think when you see the square is a big matrix of connection strengths. And the light yellow colors are low connection strengths, and the dark red colors are high connection strengths. And so these blocks would be the modules that Steve talked about in the morning. And so you take two networks, and it's important that the genes, namely the rows and the columns of the matrix, are the same in the two networks. But the connection strengths are different in general. You take these two, and the first step is to calibrate them. And this is, again, an advantage of weighted networks, is that making networks comparable or calibrating them is a relatively simple task. Okay, and so we want to find the consensus similarity, which means that two genes will be strongly connected in the consensus network, which will appear here in a little bit, only if they're strongly connected in both networks. So a very simple solution is to just simply take a component-wise minimum, or if you were to run R, you would use the function pmin. As input would be these two network adjacencies, and the component-wise minimum just means that for each component, you take the minimum of this value and the corresponding value here, and you put it in the right place in the resulting matrix. And so you get the consensus similarity, and then you can turn it into a dissimilarity and feed it into a clustering method, for example, like hierarchical clustering. So this works well for 2 or 3 sets. But if you have if you have 4 sets or more, then the minimum just becomes too restrictive, because for many pairs of genes, there will be at least one data set in which something went wrong and those genes are not correlated. So if we have more than 3 sets, 4 sets or more, you would, instead of a minimum, take a suitable quantile. So it's still component-wise, but instead of a minimum, we have a quantile, for example, a quartile, so the 25th percentile. And the corresponding R function that you would use would be the P quantile function. And again, you get the consensus network like this. And then you feed the consensus network similarity or dissimilarity into a clustering function like the hierarchical clustering. You get a dendrogram, and we get nice consensus modules. So that's, in a nutshell, that's the method. Now, if you want to get consensus modules in R, and you're using the WGCNA package, then you would use the function Blockwise consensus modules. I'm not going to tell you very much why it's Blockwise, but it's a consensus module function. Steve mentioned the Blockwise aspect in the morning. Where is my pointer again? Whoops. Sorry. Oh. Here we are. Okay, there we are. So as input, you have to give it expression data, and since you have multiple data sets, the expression data should come in a multi-set format. Again, this is something special to WGCNA, and you can see it in tutorials. There are options for splitting the data into smaller blocks. If your data is very big or your computer doesn't have that much memory, you may want to consider doing that. You have a lot of network construction options for constructing individual networks. The calibration options, how to calibrate the individual networks to one another, and the consensus quantile that I mentioned before. And what it gives you, the function, it'll give you the consensus module labels, it will give you the gene clustering tree, or if the data was split into blocks, there will be several clustering trees, eigengenes, and other diagnostic output. And if you're interested in an introductory tutorial, we have one at our webpage dedicated to WGCNA tutorials. Again, this URL will come up at the end of the talk, so don't worry about it. Now, later today, I believe Steve will talk about module preservation statistics, and a question will arise, what's the difference? When you construct consensus modules, you construct modules that are present by construction in every data set. When you study module preservation, you ask whether a module that you found in one data set is present in another data set. And so the two methods sort of give different answers and answer different questions. So the consensus modules are really best suited to the case where you have several data sets that you want to treat on the same footing. You want to sort of pull them together and create a result, and then be able to say that each of these data sets contribute to the results in the same way. Since Steve's going to talk about module preservation later, I'm not going to go into more detail here. Instead, I'm going to show you an application how consensus modules can be used to study lung cancer. So what data do we have? There are 8 publicly available lung cancer data sets. There may actually be more, but these 8 are the ones that we used. There are 4 independent data sets that are described in this publication by Shedden et al. I lost my mouse pointer again. There's another one by Build. These 5 data sets were measured on Affymetrix platforms, but not the same. And then there are 3 Agilent data sets from 3 different groups. Now, why to do consensus modules here? One reason is that the concordance between the data sets is very poor. And it's difficult to find genes that consistently relate to survival time. And so maybe consensus module analysis could help. Now, if you have never worked with lung cancer, or even if you are an expert, you should know that lung cancer, as many other cancers, comes in several different subtypes. And so the different subtypes are very different from at the molecular level. So it's hard to kind of pull them together and study them all together. So we restrict the analysis to adenocarcinoma here. And since we have 8 sets, we don't use the minimum-based consensus modules. We use the quartile. So here is the dendrogram, the gene clustering tree of think it's about 9 or 10,000 genes across these 8 sets. And you can see that there is not that much that the 8 sets have in common. There aren't that many branches. Actually, most genes kind of look like their background, and that's because there's not that much concordance. But we did find 5 modules here, and this heat map below the dendrogram tells you how each gene relates to survival time. So where you see blue color, those are genes that are downregulated in patients with short survival time. And where you see red, like here, those are genes that are upregulated in patients with short survival time. So if you look very carefully at that figure, you will see that this blue module here seems to have mostly red genes under it. So it looks like we may have found a module that consistently is upregulated in patients with short survival time. Of course, if you use, if you want to be more quantitative, if you want to see what the strength of that association is, you can calculate the module eigengenes for each of these modules. I think I lost the cursor again. Sorry, here we are. So you calculate the module eigengenes in each of these data sets, and you relate it to survival time, roughly speaking. And indeed, the blue module is weakly but consistently related to survival time, so that the meta-analysis p-value is actually pretty strong. It's 6 times 10 to the minus 7. And that blue module turns out to be very highly enriched in cell cycle genes, which makes sense. It reflects the known biological fact that a fast proliferating cancer indicates a poor prognosis and a short survival time for the patient. Yeah, sorry. Let me, yeah.
What are the 5 in the meta-analysis?
Sorry?
The 5, number 5 for the P1.
Oh, the 5 is a Z statistic.
Oh, it's a different.
Z statistic, yeah,
Okay.
yeah. So the Z statistic is 5 and the corresponding p-value is 6 times 10 to the minus 7.
Okay, but for the other columns?
Okay, for the other columns, they list, so the upper number is the Z statistic and the lower number is the p-value for the association of that module with survival time in that particular data set.
But with the Z value like 0.37, 0.36,
Yeah?
the P1 is 0.005.
Oh, no, I'm sorry. These are correlations. I'm sorry. Yeah, I was wrong. I'm sorry. Yeah, these are correlations and p-values. So for the meta-analysis, we have z-score and the p-value. I'm sorry. Yeah. Any more questions at this point? Okay, so at this point, we have a module that relates significantly to survival time, so that's great, but what can we do more with it? The challenge across these 8 data sets is to pick genes, is to pick biomarkers, if nothing else, for survival time. Okay. The other challenge is to identify functional categories that associate with survival time. And so one way to find sort of the biological categories or pathways or processes that might be associated with survival time is to use a standard approach, meta-analysis of gene association with survival time. And the meta-analysis gives you a ranking of genes, and so you select a number of top genes, and you look at the enrichment of that top number of genes in known categories. And so what happens is, when you do that, in this graph, I'm showing dependence of the enrichment score, which is just minus the logarithm of the p-value, as you increase the number of selected top genes by meta-analysis. Just leave out the marginal. Okay. Now, you can also study the enrichment of genes that have the highest connectivity in the survival-associated module. So we have the module, and we have a network, so we can calculate the connectivity, intramodular connectivity of each gene in that module, and pool the information from all the 8 data sets. That gives us a ranking, and we again select the same number of top genes, and we see what the enrichment is in the cell cycle. And you get a much higher enrichment, which means if you look at the consensus module, you will get a much cleaner functional annotation than you would get from just marginal meta-analysis, meaning meta-analysis without taking into account any of the network information. So I want to show you another application, which has sort of another point of view or another slant on consensus modules, which is a consensus analysis across 4 brain regions in Huntington's disease patients and controls. So what kind of data do we have here? We have data from Huntington's disease patients and controls. The data were published by Hodge et al. In 2006. Maybe I should say a few words about Huntington's disease. It affects primarily motor skills. The biggest changes are observed in the caudate nucleus and much smaller changes in cortex and cerebellum. And it primarily affects neurons. It basically causes dying of neurons and a corresponding increase in a sort of an inflammatory response in the brain, which means the increase in astrocytes, oligodendrocytes, and microglia. And so here we want to do a consensus analysis of the 4 data sets that Hodges et al. Measured, namely the caudate nucleus, the motor cortex, association cortex, and cerebellum. And if I just told you that the Huntington's disease primarily affects motor skills, and you know a little bit of brain anatomy, which some of you know much more than I do, you would say, well, probably the caudate nucleus and maybe the motor cortex should be the most affected by the disease. Okay, so here's the consensus dendrogram. And again, there are 2 sort of big branches here and 1 smaller branch. And when you look at the heat map of the association of individual genes with disease status, you will see that these 2 big branches have something to do with the disease. Namely, this branch with the blue color is underexpressed in HD patients, and this branch here is overexpressed. And so, if you have just heard that Huntington's disease causes the dying of neurons and a corresponding increase in astrocytes and oligodendrocytes, then you would think that, well, maybe this corresponds to neurons, and this module or group of modules corresponds to the inflammatory response. And that is really the case. So here is a heat map that a heat map of the association between the module eigengenes, here are the modules, and the Huntington's disease status in the 4 areas. So these are, each column is 1 brain area. So if you look at the red modules, what are they enriched in? Astrocytes, microglia, oligodendrocytes. The blue modules, the ones that are downregulated downregulated, or their expression is down in Huntington's disease patients, their neuron, this is a synaptic function, which is also related to neurons, down with Alzheimer's disease, that also probably corresponds to neurons. And if you look at the intensity of the colors, you will see that, yes, the caudate nucleus is probably affected the most. The motor cortex may be affected slightly less. And then the association cortex and the cerebellum are affected less again, but they are still affected. The effect is pretty consistent across all 4 regions. So, so we have the consensus modules. So we know that there is, the consensus modules reflect basically what is common between those 4 brain regions in terms of the organization of gene expression and the response of the gene expression to the disease is broadly similar across all 4 areas.
Quick question.
Yeah?
So if you go back, it looks like then from what you said, it wasn't so much a problem here, but if it was the case that there was signal in 2 brain regions and much less or no signal in the other 2 brain regions, then does the consensus approach would it mask the signal that you're trying to find?
It might, yes. Yes, it could. It could happen. But the consensus modules, the condition for finding consensus modules is not that there is a relationship to disease in every data set, but that the organization of the gene expression is broadly similar. So that's another thing that I wanted to show you here is that the consensus modules can be used to build meta-networks. Namely, you take the eigengenes of the consensus modules and you correlate the eigengenes. And you only have about 10 eigengenes per data set, or maybe 20. So instead of 20,000 genes, you have 10 or 20 eigengenes, and you create a correlation network from these eigengenes. Okay? And because you have 1 eigengene per input data set, you then get, in this case, 4 different eigengene networks across the 4 data sets. And you can study whether these eigengene networks look broadly similar, namely whether the sort of the large scale or bird's eye view of the network remains similar across the 4 data sets. So I'll give you a little bit of math, but if you're too tired after lunch, you don't have to worry about it too much. So the eigengene network is simply defined as a signed network with power beta equal 1. So the adjacency is simply 1 plus the correlation of eigengene I and eigengene j divided by 2. And then you can define a preservation network. Preservation network measures how much the eigengene correlation is preserved or varies among the data set. And so if you have 4 input networks, for each component you would take the maximum of that component across the 4 networks and subtract the minimum. And that sort of gives you, that's high if the correlation varies a lot. But for a preservation, we want the opposite. We want to be high when the correlation varies only a little, so we subtract it from 1. That way, the preservation is high if the maximum and minimum are the same, basically. Okay? And the mean preservation then just measures the overall preservation of the eigengene network, and it's just the mean of the preservation network. So the mean preservation is a single number. It does not have components, ij. It's not a matrix anymore. So here's what happens if you apply this eigengene network and preservation methodology to the brain regions. And for clarity, I only show you the caudate nucleus and the association cortex. So in principle, this is a 2 by 2 matrix of plots. In principle, since we have 4 regions, we could have a 4 by 4 matrix, but you wouldn't be able to read it if I put it up on a transparency like this. So let me just show you 2. So here's the eigengene network in the caudate nucleus. And if you remember the heat map here, these modules are the ones that are that go up in patients, and the bottom ones are the ones that go down in patients. Okay, so the ones that go up with in disease patients, they're highly correlated. The ones that go down form this block, they're highly correlated. And the 2 blocks are anti-correlated. So the eigengene correlation is broadly negative between the 2. And then there are some modules that are not really all that correlated with anything else. If you look at the association cortex, the ones that were going up, namely the inflammatory response, the astrocytes, oligodendrocytes, microglia, they still remain pretty highly correlated. But the neuronal modules that were pretty highly correlated here, they're not as highly correlated in the association cortex. And that's reflected in this preservation heat map. So you notice that the preservation heat map is basically all red. So overall, the networks are pretty highly preserved. That's a good sign. That just means that we're not finding something random that you change your data a little bit, or you change the brain region a little bit, and suddenly the entire network changes. That would not be very good. So the overall preservation is 0.88. That's pretty high. But there are some slightly lighter areas around here, which is where the neuronal modules are. Okay, so this methodology basically lets you look at the network of 20,000 genes from really far and just see, in this case, I think it's 10 or 10 eigengenes, get a reduced view. And in this case, we did not discover a cure for Huntington's disease. This hasn't been super useful yet in terms of really discovering the underlying biology or cure for Huntington's disease, but it certainly provides at least sort of a food for thought for biologists. It provides something that we can go to the biologists and ask, does this make sense? Is this something that we can push forward? Is this something that we can mine for further information? Yeah.
If I understand correct, here you calculate the meta eigengenes based on the consensus between the two networks, right?
Roughly speaking, yeah.
Then you use it to use this constructed, meta eigengenes to say you preserve similar pattern between the two networks.
Mm-hmm.
It's kind of cycle.
No, it's not really. Because the consensus modules mean that the genes, see if I can get the pointer up again. Here we are. The consensus modules mean that, for example, genes in module 8 are correlated in this data set and in this data set. It also means that genes in module 5 are correlated here and they're correlated here. But the correlation of the module 8 with module 5 could very well be different between the two data sets. It doesn't have to be the same. Okay? If I tell you that module 8 is oligodendrocytes and module 5 is neurons, it kind of makes sense that neuronal genes go together, because if you get more neurons in your sample, then the expression of you will measure a higher expression for neuronal genes. But it's not necessarily true that oligodendrocytes and neurons are anti-correlated. You could imagine situations where they are not correlated at all because there's something else going on. So in this sense, it is not, I wouldn't say that it's redundant, that it's circular or redundant, or we're setting up the situation such that we should observe a high preservation. So I'll just finish with a little summary slide. So from the cancer application, I try to show you that the consensus analysis can, first of all, identify a module that consistently relates to survival time, which is in general difficult. And second, that the module provides a much cleaner biological interpretation than a standard meta-analysis would do. And for the Huntington's disease application, the consensus module analysis shows that the Huntington's disease affects different brain regions in a broadly similar manner, but it also shows differences in the way the regions are affected. And then if you study the eigengene network, the consensus eigengene network, it gives you sort of a way to gain insight into what the commonalities and differences are in the network organization from a sort of a long distance view, a very coarse-grained view, very much reduced view. So I'll just finish with the references. The consensus modules and eigengene networks were published in the paper on top, and the cancer application of consensus modules We published it recently in this paper, and the Huntington's disease is still work in progress, so that will take some time to finish.